So my name is Caitlin. This is Behind the Scenes of Science, a program from the Allen Institute to help students like you learn about different career pathways in science. So this is a Zoom webinar. We can't see or hear you, so please use that chat to chat with each other, and make sure you put any questions for Jocelyn in the Q&A button, which is down in the footer of Zoom, and it looks like two speech bubbles. So the goal of this webinar series is, again, to give you a sense of some career pathways in science. How do you get from being in high school to being a professional in the world of science? And what does the day in the life of a scientist look like? So I'm going to start with some questions to get us going, and then we will open it to questions from you. So we will have plenty of time for that. Please drop those in the Q&A as you think of them. So to get us started, Jocelyn, can you just introduce yourself?
Hi, I'm Jocelin. I am a research associate at the Allen Institute for Brain Science, and I've been here for 7 months.
So what was your science journey from high school to now, to being a research associate?
It was pretty disjointed, actually. So in high school, I took pretty much every class I could get my hands on. I did a lot of art and philosophy, language arts, science classes, and I thought that I was gonna be a philosopher. And so I went to school, or I applied for schools, applying to both science programs and art and philosophy programs. And it happens to be that the school that I really liked when I visited didn't have an art program, and that really derailed my life plan. And so I was like, well, I'll go for biochemistry, but if it doesn't work out, then I'll switch schools, not a big deal. And then I ended up really liking it. And I realized that the career paths weren't quite as segmented as I thought they were. So there is a lot of art and philosophy that's practiced in science. And so I was able to get my fix kind of in that field. So I went to Seattle
college was this?
Oh, Seattle University, yeah. And then I got a bachelor's of science in biochemistry in 2016. I joined a emergency vet clinic as an emergency triage nurse to try and get like clinical experience. And I was like, if I'm gonna get clinical experience, I'm gonna get it as fast as possible. And then waffled between, do I want to be a lab technician? Do I want to be a veterinarian? Do I want to go to grad school? Did that for about 4 years, and then decided that research was the direction I wanted to go in. And so I ended up here, and I work in the Histology Lab in the Brain Science division.
Before we get on to what an average day looks like, can you just tell us very briefly what histology means?
Yeah, so pathology is the study of anatomy that's not doing what it's supposed to, like a diseased anatomy state. And histology is an examination of those pathologies. So it's pretty much any time you have a surgery and they cut into like what they've taken off, the histology lab is looking at that and getting those samples processed so that a doctor can read off and give information on treatment.
So the Allen Institute isn't a clinic, so you're getting an echo. The Allen Institute isn't a clinic, so for you, histology is being used for research.
Yes, which is very, it's actually a relatively recent transition that it's been called the Histology Lab. So our lab has been, is like a support lab for all of the other branches. And so it's been called 3 different things. It's been called the Brain Atlas Lab, the Production Lab, and now the Histology Lab. And we're called histology now because of a lot of our work has to do with single cell characterization, which is where we do like a bunch of really cool antibody tests to find out as much information as we can about one cell. So it isn't a clinical setting, but it's kind of it's very unusual for histology to not be in a clinical setting. A lot of our training is in the context of human clinical pathology.
So when you were doing this, what does an average day in the life look like?
It's a lot of lab work. So we have 4 main pipelines that we help on. So there's they're called the Biocytin pipeline, the individual cell characterization pipeline, the mesoscale pipeline, and then reagent prep, which pretty much means that we're kind of like a central hub. If researchers want to, if they want to get data, they design everything from like selecting what genes they want a mouse to have up to reading the data itself. And so those are handled by other teams, and the in-between is handled by us and then several other labs. So it's a big collaborative project. Generally on a day, we have a whole manager who's just she's a lifesaver. Her job is coordinating all of these different conversations. And so she assigns us tasks on a service that we just log into every morning. And then we look at it and we say, okay, I need to stain these types of grains. I need to stain it with a certain stain. I need to prepare a bunch of ingredients for that. And then also prepare ingredients for other labs. So usually the morning is taken by just doing really precise calculations and like mixing things together for other labs. And then the afternoon, we support the mesoscale pipeline by taking brains that have been sliced into really, really small pieces and then ordering them from front to back. So the afternoon is, it's nice to do in the afternoon because it is like very repetitive and it's kind of soothing. So morning, a lot of chaos, a lot of organization. And then the afternoon, we kind of just support other pipelines by doing some like fine detail work.
We have a follow-up question from the audience about that. Is your work very hands-on?
Very, very. I was actually pretty surprised. So I was watching some other videos from this series, and then I was looking at other people who have my same job title, and I was like, oh, data analysis. I haven't done any data analysis, my understanding is that I've joined during a very busy period, and so we are actually producing a lot of data. So in times like for COVID, when you have to work from home, we would do a lot of data analysis. But right now, our priority is coordinating everything and making sure that everything gets produced. So we will worry about the data in a few months, and I haven't seen that part of the job yet. So my experience is that, yeah, it's 8 hours a day, hands-on science.
Yeah, and as Jocelin mentions, we have had other people speak in this series who their entire job is data analysis. They're hardly in the lab at all, if even a little bit. Some of them are purely data analysts. Mm-hmm. So what does a scientist day look like can really vary. So you've spoken a little bit about the variety in your job. What is your favorite thing about your job?
My favorite thing, I think, is the problem solving. So we are given, so it looks like we're given recipes when you're given like a chemical solution to make, but there is a lot of troubleshooting that goes into it. So you have to have a really good understanding of the chemistry of why reactions work a certain way. So you can follow a recipe, make a solution, and then test it, and then the test just tells you like, oh, this is wrong. You need to figure out where you went wrong. So there's so many factors that go into making the stars align, so you can have a research that produces really accurate data. So it's a lot of troubleshooting and just making really, really precise movements, and also a lot of documentation. For example, right now we have one of the things that we make is paraformaldehyde, which is a fixation agent that we use for the mouse brains to make sure that they're perfectly preserved when they arrive to us. So one thing that we're working on right now is that there was a sample from a 3-day period where the brains didn't stain as well as they could have. And so right now we're going back at all of the documentation from this preparation back in early December. And it looks like what happened is that the heating plate had been a little bit uneven. It had dropped in temperature and went back up. And that could have affected the chemical composition of the solution that we provided to that team. And then it didn't work as well. So you have to have a really good understanding of how the science works and be able to go back and see where things could have gone wrong, and then correct it for the future.
That's some great detective work to have narrowed it down to what caused that.
Yeah, and me and my teammates make really good notes on everything that we do for this exact reason, because we want to produce the best science that we can. And if you make good notes, then you can catch mistakes like that.
Yeah, that's a really great example of how lab work, like the facets of lab work that aren't just, you know, actually at the bench interacting with your samples. Yeah. Well, so sometimes, as you mentioned, science does not go 100% smoothly. What has been your best science failure? Something that was a good learning experience, funny, surprising?
Actually, I think that my biggest science failure would be that I thought that I had a very rigid idea of what science was, and that was kind of based on like the chemistry classes that I took in university. So I was like, oh, it's boring. You have to like write in lab notebooks and just be super precise. And like I didn't pursue a career in lab work for several years after getting my degree because I thought me having worked in one lab was enough of a worldview for me to know how all labs worked. And so I think my biggest mistake was thinking that science is a monolith. And as you can see, if you watch the other talks here, I mean, there's incredible variability just within the different groups within the same overall team. So there's a lot of directions that you can go in, and there's a lot of flexibility.
Yeah. Well, we have plenty of time for questions from you, our audience. While you get those queued up in our Q&A panel here in Zoom, Jocelin, you want to talk about some of these cool scientific images you sent me?
Oh yeah, of course. I went way too fast through the day explanation. So I wanted to get into the pipelines that we support because I think they're really cool and interesting. So again, those pipelines are the BioScience pipeline, the individual cell characterization pipeline, and the mesoscale pipeline, and then the reagent prep. The reagent prep is like all clear liquids, so I don't have any really good photos of that. But the biosite prep, it's really exciting being able to be involved in so many projects because we help support several points of this pipeline. So the reagents that we make at the beginning of the day go to a different team. And then that team takes a brain slice and then puts it in that reagent to preserve it. And then a different team will take recordings on each end of a neuron, and then they'll record the electric potential between the 2 points. And then they'll inject a dye into the neuron, and then we stain it so that the dye can be amplified and can be seen under a microscope. So we would get the very beginning with the reagent prep, we'd get the middle with the staining, and then at the very end, in the next image, in our downtime, we also do neuron tracing on this open source program called Mosaic. So that'll be the next image. So Mosaic is a citizen science project where we take the sections that we've stained, we give them to imaging. Imaging processes them into a huge multi-layered image, and then they open it up to the public. So you can go in and trace all of the anatomy of the neuron. And what that does is that we compile that information over and over and over again. And then neuroanatomists and data analysts can go in and say, oh, we have all this data from the beginning, the middle, and this endpoint. Now we can compile them and then learn more about this neuron. So it's really cool that we can see all steps of this process. And we also can see, we can just see how much goes into to procuring a sample, to getting the data, and then eventually to a paper. The next project that we have is the individual cell characterization, and that's where like a lot of like the quote unquote histology work is. Otherwise, I think we would just be like a general biology lab. So the individual cell characterization is a pretty cool subset of histology where we stain individual cells based on proteins that we are anticipating are going to be in the cell. So my understanding is you guys learned about ELISAs recently. So ELISAs are a test where you react if you're looking for something in an individual well, and then you put antibodies in to have a potential reaction with it. And if the antibodies do react with what is in the cell, it means the original, like the analyte that you were looking for is confirmed to be in there. So we do that, but we also look at, we can make further characterizations about structure based on like the patterns that appear when we stain something with an antibody. So that's really interesting to us because we can, so the first thing we can do is that we can look at the actual anatomy of structures that we know are in the brain. But since every single mouse that enters the Allen Institute has a known genome, we can also look at their genome, say, oh, I see this should encode for this, and I expect it on like the outside of the brain. You look on the outside of the brain, but you also see it other places. And so you're like, oh, I need to further investigate why the thing that I thought was only on the outside is also on the inside, and then whether it has a similar function. So it's a really good, it's just a really good way to build like foundational science because our ultimate goal here is to build a database that other people can access and use for their own projects. Oh, sorry, I forgot about the Mesoscale Pipeline, which is actually one of our bigger ones as well. So the end goal for research is, for most people, I guess, except for like evil scientists, is to improve our medical, like our medical capabilities and to know more about our brains in general. So we have like functional neurobiology where we see things like EEG readings or even behavior. And then we have all the way down to individual cell characterization, which is what we do with like those specific stains. And then the in between, there is large gaps in our knowledge on how this extremely small and the extremely large data points connect. And so that intermediate we refer to as mesoscale. And so our goal is to build the database that connects these very, very small data points, these very, very large data points. And so that uses like electrical and anatomical and other imaging modalities to gather as much information as we can about those intermediate steps so that we can draw better conclusions about when we see something on a molecular level, what it means for like a clinical level and vice versa. There is also the Mesoscale project is a continuation of the Brain Atlas, which is what my lab used to do 100% of the time. So the next image, I believe, should be of the 2D Brain Atlas and the 3D Brain Atlas.
Yeah, so I have the 2D Brain Atlas first.
Beautiful. So what we do as part of Mesoscale is when we're given all these brains to sort from beginning to end, it's because we're adding that data to this 2D Brain Atlas. So we're given a single mouse brain. It's cut into about 140 pieces, slices, and every mouse brain is cut in the exact same way. And so when we order them, we know based on the genome of the mouse and the some preliminary imaging that we had done before, that there are certain regions within the brain that will react with the staining that we use, and we want to know more about that. So we find these markers, and then we do special staining on them to learn more about the marker or to get better data on the markers. And then we compile them into this 2D atlas where you can scroll through and see each individual brain slice. But the next step is going to be the 3D brain atlas, where you aren't just moving through it image by image, kind of like a CT scan. You're able to connect data points in a 3D space, and they refer to it as kind of like brain GPS. So if you know, so the ultimate goal would be if you know of a structure in the brain, you can immediately navigate to it. And then the next video would be a
Yes.
simulation of the 3D brain atlas.
Yeah, before I jump off of the 2D brain atlas, I did want to mention that the left side of this image is a stained brain and the right side has been illustrated to show those structures.
Yes.
So the right side is an illustration.
If you ever want to, so the this 2D brain atlas is what's called a coronal section. So it's not that typical like big spongy cute image of a brain that we kind of think of. It's if you're facing me like this, if you take slices going back, it's a coronal or the crown image. So if you go through, you can look from nose to back of head to see the structures of the brain from the front on view.
All right, here comes the 3D.
All right, and then this is a progression through the brain. So you can see all the structures, all the functional structures. And the goal is when we gather all this data is to have a better understanding of neuroanatomy and how that relates to function. But then it's also to understand what is where and then how they interact with each other. So the red points that show up on here are the points that we're gathering with our mesoscale data. So the way that we do that is the gene editing team at the Allen Institute identifies a mouse that has a gene of interest, and then they inject the mouse with a virus that contains a fluorescent tag on it, and it connects to the region of interest. And then once the mouse has been sacrificed and we have the brain sample, we can do stains on it and we can put it under fluorescent light and then find the places in the brain where that genetic tag has been placed. So that just gives you more information on anatomy, and it gives a lot of flexibility for researchers to really ask questions that they had never been able to ask before, because they're able to have a lot of flexibility with where with what they're looking for.
So we have some questions from the audience that we're going to go through now. So how did you know where you wanted to go to school and what you wanted to study? So you talked a little bit about that you chose this school and that it didn't have a program you were looking for. How did you choose?
So I'm not proud of this. I did not know what I wanted to do. So I think that because they make you choose a college so young, I don't think I knew who I was as a person at 16 or 17. And so I think I knew that I would be happy with many career paths. And I knew that I wanted to learn more than I knew then. I had done the International Baccalaureate program in high school, and I just knew that I was scratching the surface of what people could know, and I wanted to be able to advance any field that I was in. And so my goal was to get into the program that I thought would teach me the most. So Seattle University, there was a lot of factors that went into it, and it might be too much information, but I knew that I only had a set amount of financial aid that would be available to me. And so I knew that me finding more financial aid would be stressful enough, so stressful that I wouldn't be able to learn enough. So I chose Seattle University because they offered me a good scholarship. And I knew that they had one of the 3 programs that I was interested in, and I was able to balance the practical needs of having enough loans to go to school with the ability to maximize my education. So that's not a very romantic notion, but I think it served me really well. And I really enjoyed the classes that I think I probably also got pretty lucky because the classes that I took were designed to be holistic in nature. And so I think that going to a school that really prioritized the interconnection of ethics and science and really respecting that you should educate the whole person and not just on an individual discipline. I think I got pretty lucky that it was as positive of an experience as it was.
Well, thank you for sharing all of that. And not every decision has to be like high in the sky romantic ideal. Sometimes those practical demands are what makes it.
Yeah. And I mean, and it's just such a big decision that I think I was overwhelmed by all the options. So sometimes you have to pick a criteria and go with it.
So you work on a lot of different projects in your job. What is your favorite project to work on?
I think the individual cell characterization is the most interesting because it has a lot of like the really nitty gritty chemical reactions that I like. I really liked my chemistry classes and my organic chemistry classes in school. And so with the individual cell characterization, you kind of get a smorgasbord of all of like these core concepts. So from the very beginning, you have to say, oh, I'm looking for gene X. And I know that gene X is expressed in mice and people. And so I need to find a mouse that naturally expresses gene X. So you get a little bit of genetics there. And then you have to be able to identify it's like a lock and key mechanism for finding a protein and then locking an antibody to it to build a stain on. And so you have to be able to have like a practical ability to like find an antibody that's affordable and reproducible and stable. So you kind of do like some detective work to uncover that, and then you have to be really good at experiment design to set up the exact conditions for to really maximize the potential of that antibody. And then you sometimes get to change things on the fly sometimes if you know your science really well, and then you see that a sample there is just like an accident, things happen, and the sample isn't as good as you thought it was going to be. You know that there's variability in your experiment design. Change things on the fly was a little bit extreme. There's variability in your experiment design, and you get to make decisions that take in all of these factors to get the best result possible. So I think that's the most dynamic of the pipelines that I work on. And I just like being able to flex all those science muscles.
So you have talked about the variety of your job. So, there's a lot of things to explore, and you've talked about the other career paths that you were interested in as you headed into college. What does the future hold? Are you interested in staying with research, exploring other fields?
Yeah, so a lot of the things that drew me to biochemistry is like the ethics of disease research. That's also what drew me to veterinary medicine, because veterinary medicine is like an emerging field where the bioethics are kind of unknown because the patients aren't people. So it's just a very interesting field of study for me. And so I really liked working here for the Allen Institute because I knew that because of the interview and because of people I talked to in the industry that they really strive to have ethical data collection. And then not only do they collect data ethically, they make it open source so we can be conservative with our resources and it can benefit the most people. So I think that I'm currently trying to train in the actual animal services portion of the brain science department, or I would go into public health, because I think that public health is also a very interesting field where you can really make changes that can positively affect a lot of people. And so it seems like the theme of my career has been like just kind of circling this idea, which I know isn't the best way to doesn't seem like a very logical way to go at like progression within a field. But I found that all of the experience that I've gained at different jobs or different classes that I took in school, it all has a cumulative effect. And I feel like I'm able to be a more well-rounded researcher by just having these varied experiences, and then also by having just wanting to work in an area where I'm able to like exercise something that I'm passionate about.
Yeah, having a career in science doesn't have to be a straight line from taking science classes in high school to majoring in science in college to maybe going to grad school. It doesn't have to be 1, 2, 3 like that.
Yeah, and if you know that you like that, absolutely go for it. And I have a lot of classmates from school who are in PhD and grad school programs, and they love it. So if you know what you want and you want to go forward, then that's the right choice. But you don't have to be perfect to be in science. You can take a circuitous route.
So we still have time for questions from you students. So if you have a couple more, go ahead and pop those in the Q&A. What do you wish you had known as a high schooler about a career in or around science, in addition to the idea that you don't have to take a straight line to go from point A to point B?
Yeah. I think I wish I had known about the variability. So I think that there is like a very, again, monolithic view of what practicing science is. It has variability like any other discipline. So you can, if you enjoy work where you know exactly what you're going to do when you come into work in the morning, you do it, you head home, you don't have any, like you don't take work home with you, and then you have like a healthy like separation of work and life, absolutely. If you want to, to work in like a startup environment where you're working 80 hours a week and like you live and breathe work, that's also very much a possibility. And I think that there's rewarding paths in both of those, and you really can make the choice for what kind of relationship you'd want with a science career. It's not restricting at all.
So you've spoken a little bit about how you have integrated your interest in philosophy and ethics into your science career. Can you speak also about how you integrate your interest in art?
Yeah, so I was actually just thinking about this the other day. So for art, I think that a lot of what I liked about art was communication. So it was taking resources that you have and then manipulating them in a way that they told a story. And I think that there's a lot of stories in what research you choose and then how you choose to carry it out. Let's see, for example, one of the research projects that's going on in the Allen Institute right now is a research project where you can go forward. They're analyzing how the brain processes a mouse approaching a picture. So there's just a really complex story in why they chose that question and how they're approaching it. And so I think it's very interesting to me that you can have like, it seems to me like a collaborative art piece, because you can have 20, 25 people all collaborating on this project, and they're all going to be asking different questions. And then they all are going to contribute a different perspective and a different problem solving strategy. And so you're given limited resources, which is that you know only X amount about the brain, and you want to know this new thing. So you want to tell the story of how that process connects. But it's just like a collaborative problem solving process. I don't know if that's quite the definition of like fine art or anything like that, but it seems like an artistic process to me.
Yeah, well, speaking of collaboration, A next question is, can you explain what citizen science is? You use that to
Oh,
talk about Mosaic.
yeah. So citizen science, being a citizen scientist is something that's really exciting to me. So it's people who can collaborate with researchers to help provide data and to provide a perspective um, that either people hadn't thought of before, or to provide like some of that data analysis that just takes time and work. So to be a citizen scientist, you just have to have an interest in the field and a willingness to learn. So there's all types of different projects. So the one that the Allen Institute runs is Mosaic. It's a neuron tracing project where we actually contracted out to a game development company, and we said, hey, we have this problem. We have thousands of images of neurons, and we're building data on them as much as possible. But tracing them takes time, and it takes skill, and we want more of it. And so the benefit of a citizen scientist is that someone who comes in and is like, takes the tutorial, wants to know how to learn, can take the tools that are offered to them through the game and then trace these neurons and offer, and then that compiled data will offer information on like measurements and anatomy that we wouldn't have been able to collect otherwise. So there's tons of different citizen scientist opportunities. There's ones like Mosaic, there's protein folding ones, there's like ecology and plant identification. There's pretty much if you see a project that you're excited about, you can Google volunteer opportunities or citizen scientist opportunities, and you'll most likely find a group who says, yes, we are happy to teach you what you need to know about this, and then you would be helping us advance our understanding of the subject.
Yeah, Mosaic is developed, the data comes from the Allen Institute, the neurons that we're tracing, but the game itself is designed by a group at the University of Washington that studies using games to run citizen science projects. So another one of their projects is called Fold It. So it's the same idea of doing research on how can we get more people engaged in that citizen science. So we are just about out of time. We have a few more minutes left. A couple of additional questions that we have time for. What do you like to do outside of work?
I am kind of a collector. I'm a hobbyist. Currently, my go-to is crocheting and snowshoeing. So I like to stay busy, and I really like that the job I have right now at the Allen Institute is really good at like maintaining a work-life balance. I've worked in jobs where that's not the case, and again, that's science. You're allowed to take any kind of opportunity that you like. So I think that I like having like a 9 to 5, going out and watching TV and crocheting in the evenings, and then Seattle's gorgeous to explore. So a lot of hiking and snowshoeing on the weekends. I also just started a project fostering kittens. So that has been really, really fun, and I would highly recommend it to anyone who feels kind of trapped inside. There are a lot of chaos, a lot of good.
How many kittens do you have right now?
I only have 2 right now. I was offered a litter of 9 recently, and I'm still debating that one. So 2 to 11 soon.
So just to wrap things up, do you have any final thoughts, things that you want to make sure to share with these students as they are moving into the next steps of their science journeys?
Yeah, I would say be kind to yourself. Science is hard. The material is just hard. Like, we're trying to push our understanding of what we know about the world, and mistakes happen. And I think just if you work hard and you have the intention of learning as much as you can, I mean, everything will fall into place after that. You don't have to be perfect. You don't have to have like a perfect research record or anything like that to be successful in the field.
Well, thank you so much. And thank you to all of you students for joining us today. I am about to drop the link here in the chat so you can watch other speakers in the series and learn more about other career pathways in science. Thank you, Jocelin, for everything, and have a good rest of your day. Thank you, everyone.